Alright folks, welcome to our new online Bio 1600 experience. This is different, I think, for all of us, but it's what we're in now. I hope everybody's adjusting and doing well to their new quarantine, self-isolation, self-distancing, social distancing lifestyle. A lot of new buzzwords and phrases coming around, but also a lot of good memes that are coming out. Like, leave it to the modern internet to take any situation and be able to put it into some memes and blast them everywhere. I hope everybody has toilet paper and doing okay on that front. If you had to beat up somebody for it, I mean, fine, so be it, right? You have to do things sometimes, but yeah, this is odd. But yeah, I hope my voice is okay. If you hear occasional songs, that's show tunes being played in another room, but for the most part, it should be pretty quiet. And so this is going to be how we go forward. So all the lectures will have this type of setup. I'll try and keep these not like, you don't expect like 3 hours of lectures every week. That'd be kind of insane. So it'll be a little different, be a little shorter. And there'll also be some companion videos for a lot of lectures. We look at animal diversity videos, so I'll reference those, and then I'll provide a playlist you can watch on YouTube. But anyway, back to business, all right? It's been a week, really, and a long week since we talked about stuff. And we started off and got into body plans. And so this is a pivotal part of it helps establish the next part of the course. We'll be looking at animals. And so these are some traits that are going to pop up. Time and time again, we're going to talk about like, oh, it has this, it has that. As we move along, the evolutionary timeline of animals. And so sponges kind of don't have a lot of this, so that's why I didn't talk about it first. But now that we're outside of that exception, we can talk about the different things of body plans, like what characteristics animals have, and like what they can vary in, like overall on a broad level. So we talked about like development, blastula, gastrula, those fun things, what a body plan is. So that was all on Friday, so check on those notes. But now, though, we're going to pick up where we left off, which is about germ layers. How fitting, all right? Of all the slides to start on, we have a virus going around. We're going to start off with a slide called germs. I just realized that. Man, sometimes timing is just perfect. But anyway, so we have a few layers, and these can vary a little bit in the groups as we look at them, so they'll definitely have some variation in those. But for the most part, you can have up to development. So animals grow, right? And so do plants, right? They have different stages, but animals have the same thing. And so you have an early stage, you grow, and then you'll become the full-on adult stage. And usually there's some differences there, with adult usually being considered as reproductively mature, while juvenile maybe a little less so. But most animals have at least one type of larval stage present, and there's many types of ways things can change. And we'll look at some of those, especially when we get to arthropods. This is a thing. And so a larva is just any sort of sexually immature form, and it's morphologically distinct from an adult. So if you look in the top right picture of the caterpillar and the butterfly and the butterfly are very distinct from each other from a morphological standpoint. One walks around with a ton of little legs and just feeds on leaves. The other one flies and looks beautiful. Very significant difference. And then eventually they undergo a process called metamorphosis, which is really what evolution is in Pokemon. That's evolution in Pokemon really just set up a lot of misunderstanding for poor children. It's really metamorphosis, but I guess evolution sounds cooler. Even though, I'll be honest, I've always preferred the word metamorphosis because it sounds so complicated. It's like, ooh, metamorphosis. It's like, oh, snap. Like, something's happening. But evolution is like, oh, okay. I mean, that's cool too. But metamorphosis, I don't know. I like anything that's got morph in it. I think it's because I read these books called Animorphs as a kid. And in that, like, they transformed into animals, so I was just like, ooh, this is exotic and crazy. And I always talk about, like, frogs and tadpoles. Like, I was like, so cool. But anyway, that's another great example of the larval form is a tadpole to a frog. Very distinct. One lives in water. One really, well, it can live in water, but it can also move on land. And then, though, once this juvenile stage occurs, this is one that looks closer to an adult, but is not yet sexually mature. And so here's a couple organisms, so a bird and a lion, These are like more juvenile stages, so they look pretty close to an adult bird, but they may be missing like maybe a couple different characteristics to them. So like usually in birds, there's a coloration change between the juvenile and the adults, and in this case we see the young male lion attempting to grow out the mane. So no female is going to confuse that with a full-on adult male. It's flat out not there. But having these different changes is important because it allows organisms to spot like which ones are reproductively mature. And so it's like, okay, they don't have to waste effort trying, court or deal with advances from a juvenile because there's no reproduction that's going to happen. It just can't be a thing. So this is a pretty evolutionary, significant little benefit here. Okay. Let's talk about body cavities. Okay. So you can have a body cavity. You could have 3 tissue layers. It could not. It just depends. But the benefit of a body cavity is it allows for you to have all sorts of just junk inside the body, right? All those crazy organs filled in there. You feel like you probably got like an extra free space in there, and you kind of do. I mean, if you have like a kidney transplant, usually they just leave your current kidney in and just add a new one in, which seems pretty crazy. You have that much spare room. But I mean, for the most part, it's jammed full with some stuff. If you ever do a dissection on an organism, you'll see that sucker is full. So same case here, but it allows for those advanced organs to come up, and the organs allow for specialization, which allow for different properties and adaptations to allow them to survive. So not bad. There's also usually a fluid-filled space, and again, if you ever cut open an animal, you'll see a lot of fluid coming out of there. And so usually it's kind of preservative at that point, but if it was still alive and you cut it open, fluid everywhere, Brody. But body cavities in this area, they also can provide a site of muscle attachment to allow things to move. And so, I mean, like, if you do sit-ups or something like that, like, there's muscles acting on that part of the body, that cavity, and allows them to actually move and have different types of ways to move inside their environments and navigate. And the body cavity itself may not be there. So you may have 3 tissue layers, you may have a body cavity. None of those things are guaranteed in all animals. We'll look at a lot of animals that don't have a true body cavity. We'll talk about the way you categorize those coming up. But also having this body cavity with the muscles, it helps protect the organs on the inside. So if we're looking at our Kung Fu Panda, dude here, the stomach kind of area and like the middle part of the body, that's the body cavity. And so that is very much protected, helps protect these internal organisms. And it can actually form a hydrostatic skeleton. So we have a skeleton made of bone, right, that holds our body together. Other organisms can have one where water serves kind of that same purpose as bone to provide that structure to them. And so that can happen and be made possible through the body cavity. All right, so bilateral symmetry. So another characteristic I mentioned when we first started body plans was the importance of symmetry. And so we're going to look at a lot of different animal groups, and we'll be differentiating between which ones are radial, which ones are bilateral. Most are bilateral, and so the last part of the course will be all bilateral animals. But this first part will have much more the radial side of things. And so that's just 2 different types. And so bilateral symmetry means that the left and right sides are pretty much mirrors of each other. Looking at the pictures, we see our gazelle type thing and the whale. And you can look very clearly and see there's a left and a right side. Also with bilateral symmetry, you have the differentiation between a head and tail end. And so this corresponds to having anterior and posterior ends. It can also be called, cranial and caudal ventral, I forgot. But there's other terms for it. And this is going to correspond to nervous development. Comes around the bilateral symmetry, how it's different. But I mean, you very clearly have a head end of an animal and a tail end of an animal. And then you also have a belly to back type of arrangement too. And that's also called ventral and dorsal. Ventral meaning the underside, dorsal meaning the back. So like dorsal fins are on the back side of an organism. There's a dog barking. I don't know if you can hear it. That dog barked like 12 hours straight yesterday. Made me go insane. If it does the same thing again today, I might die. So anyway, so with bilateral symmetry, one of the things that you see with that is cephalization, which is the building up of more of a head-like region. So you have like a brain type thing forming. And so if you have a bunch of nerves that collect near the anterior end of an organism, well, that builds up in this way, and so you have this head type region. And so this helps you like really just kind of navigate in your environment. So like you know which way you're going. You move in one direction. You can be aware of things going on in that section. It's just kind of just a pretty overall solid adaptation. And then bilateral organisms are the only ones that can have a mesoderm. So that's a pretty big distinction. And then they are generally more active and they move actively because like they have a concentrated head region and they have accumulation of nerves in an area, allowing for a central nervous system to develop because it doesn't be scattered throughout the body. And we'll see how this can vary and be different in a radial animal in our next animal diversity conversation. So that's very much a thing. And in terms of like the genetics of bilateral symmetry is extremely hard to break in animals. So it's very well conserved in terms of genes. And so if you have any sort of deviation from it, like you just die and don't develop. So this is very heavily conserved and it's very tough to break. So it's strong. It shows how strong this is from a genetic standpoint. Okay, and some animals actually will use like quality of symmetry and when selecting mates. So I know in barn swallows that could be the case. Look for like how nice the symmetry is. All right, so here's a couple of examples of ways we're not perfectly one-to-one symmetry on the right and left, right? So this is a picture on the left of the human internal organ system, and obviously it's not perfectly even, right? The heart is more on the left side than the right. When I say left and right, I'm not getting that confused. I'm using the anatomical positioning to where the left side is always on the person you're observing, not your own personal viewpoint. But also the liver leans more on the right. The intestines kind of vary a little bit. So, you know, there's differences. And then, of course, we have the flounder. Nature's oddball. A very unusual characteristic they have. This little flat fish specimen here where the eyes have actually moved to both be on the same side of the head. It's a tough thing to do, but they pulled it off. And it does fit with their lifestyle. So like they're technically considered bilateral animals, even though like both eyes are on the same end. That's just an exception, but they're still bilateral symmetry in more ways than one. The dog has to stop barking for now. Oh, please stop barking. Anyway, so body cavities, right? You can have them, you cannot have them. But what is the range here? What are the terms? The true term for you see pop up in science for a body cavity is coelom. Does it have a coelom? And that it comes from the mesoderm itself. So, bam, you have mesoderm, you can have a coelom. And you can have organisms that show all sorts of variation here. You have an acoelomate, which is one that does not have a coelom. And the common one there is a flatworm. You don't know what a flatworm maybe is just yet, but we'll look at their group soon. Platyhelminthes, the name of that group. Then you have a pseudocoelomate. And this is one where it has a coelom, but is not 100% lined with mesoderm. And so a nematode or a roundworm is an example of that. There's diagrams that come up in the next slide you'll get to see that have this. And then of course we have coelomates. These are the real deals, the ones that have a true coelom. And so it's fully lined with a mesoderm. And then an earthworm is the example there. And right away you see how worthless of a common name worm is. Three very different characteristics on three things supposedly called worms, but they have not a whole lot of closeness to them. So very, very different. But these things were abandoned a long time ago. So this used to actually be a way you could break down and talk about, like from a phylogenetic standpoint, a systematic standpoint, these are groups. And so you have the acoelomate phylum, the pseudocoelomate phylum, and the coelomate phylum. But after further DNA analysis, they realized that wasn't the case. Like, that's not really breaking these things down in the correct way. And so now we use a different set of categorizations that don't use these as terms. Instead, they're just more like casual terms like, oh, this is an acoelomate. You know, it lacks a coelom, but it does not speak to its evolutionary history. So that's a big difference. So we'll look at some of those. And so next up, here's a picture of the different types of organisms, showcasing the different types of coelom they can have. And so we have our flatworm up top, the Platyhelminthes. It's kind of cute, has little eye spots. And then, I wonder if you can see this mouse. Probably not. But that's there, and so you can see it does not have it has, a mesoderm type area, but does not have a coelom there. So it just has a region with a tissue there, but there's no body cavity. Then the pseudocoelomate with a roundworm, you can see that it has a body cavity, but the mesoderm is only on one side. It isn't found around the endoderm as well. Meanwhile, if you look at the coelomate side with our little earthworm friend, there you can see a body cavity is present, and then you also see the endoderm is fully enclosed by, or fully covered by the mesoderm, and that all aspects of the coelomate, no matter where that body cavity is always in contact and covered by mesoderm. So that's how you can tell the difference between them. All right, so when it comes to these type, another thing with animals is evolutionary development, like different pores. So in the beginning stage of development, a pore develops. What does that become? So the blastopore, does it become the mouth or does it become the anus first? And usually you'll have a case where the other one will develop along the way. It's just which one comes up first. And so if in the right, you see the diagram. So right, you have fertilization occurs, then the dividing, cleavage, the hollow ball of cells, right, that whole thing. But then you see the blastopore pop up. What does that thing become? And so if you think about it, like the mouth seems pretty important, but I mean, like, hey, the GI tract, it's important to have, both spots there. And this may surprise you, but the mouth develops first, not in humans, but in worms, mollusks, and arthropods. So our nautilus friend here would very much have its blastopore become the mouth first, not in humans though. What worms? Excellent question. Worms works in general because they all fit the same pattern. And then arthropods, of course, so like the insect world, they all have this. Something else that protostomians have, which that's what this is called. So if your blastopore becomes the mouth, you're called a protostome. They also usually have a different type of cleavage called spiral cleavage, and it's also a determined type of development. So like what those early embryonic cells will become is determined very early on in development, and this is different than deuterostomes, which is what we are. And they have thought to have separated out from pathways like over 500 million years ago. So there's a significant split here between the protostome path and the deuterostome path. And then there's our deuterostomes. And so, yes, us, mammals. This thing down here in the bottom left is a tenrec. Cool little animal. Check that thing out. Weirdo face. We had a skull of it at one point, but I don't remember seeing it when I was looking at specimens this week in lab. But in this case, the deuterostomes, the blastopore, is going to go anus first. Then the mouth forms later. So you form that forest tract, and then the pathway will continue on and to form the mouth at the end. So echinoderms, or sea stars and sea cucumbers, those friends. And then the chordates, which are us, birds, amphibians, reptiles, fish, all those, and sharks, all those types of things. And so we have radial cleavage during development, and then we have indeterminate cells, so they can have the capacity to develop into almost anything. So it's a lot different as a process. So yeah, okay. Those, but when you're looking at the bilateral groups of animals, so bilateral symmetry animals, which is what those deal with. Sorry, I don't think I really mentioned that. The ones that show bilateral symmetry are grouped based on protostomes and deuterostomes, where that blastopore becomes. And there's three groups in this. So you have the deuterostome group, the ecdysozoan group, and the lophotrochozoan group, the big three. Ecdysozoan is characterized by the shedding of some sort of like external shell that they will shed. So the process of ecdysis is molting. And then you have Lophotrochozoa, which, oh man, that's a fun one to say. I encourage you all to say it with me a few times. Lophotrochozoa, Lophotrochozoa, Lophotrochozoa. But that thing is characterized by having a lophophore and a trochophore. So a lophophore, which is a feeding structure, and then a trochophore, which is a larval form. But that was grouped together based off of those characteristics all those members have. But deuterostomes includes hemichordates, which are a weird kind of worm, called an acorn worm, the echinoderms, sea stars, and all that sort of stuff, and then us. So it has vertebrates and invertebrates to it. Meanwhile, the ecdysozoans and lophotrochozoans are all inverts. So this is a big deal. So this is a huge shift in characterizing this, the life, the systematics of life, the animals, compared to just basing it on coelomates, acoelomates, pseudocoelomates. So the guy who was responsible for this, or a big player in it, was none other than Ken Helanich, the guy who mentioned earlier, shaking the game up 20 years later when it comes to cnidarian or ctenophores and sponges. And now that same guy, he's made his his bones before and shaken up the tree of life and how these things are broken down. So, the traditional one was broken down into Duodenstones, protostomes, and then, all that sort of thing. And then now it's like, forget that, man. Like, we're going down into it. We're going to have Duodenstomes, Lophotrochozoans, Ecdysozoans. So the man has changed the game before. And so it's a big difference. It's a cool deal. And so this is the old tree based on the physical structures. So you can see it's broken down by coelomates, pseudocoelomates, and coelomates, and also protostomes and duodenstomes. Now, though, very different. You still have the protostome-duodenostome comparison, but this is more based on molecular data, and this is what we use now. And so you can see those Lophotrochozoan clade, the Ecdysozoan clade, and the duodenstomes, off on their own separate little path. So this is what we use now, and this is the tree we're going to reference the rest of the semester. So we've already talked about Porphyra, our sponges. We're going to get into Cnidarians soon. And this is also a great tree because it shows you various traits about that along the way. And so we can see, like, boom, multicellularity right away. Then tissues came next. And so, yeah, pretty cool. See where bilateral symmetry pops up. So again, most animals. All right, and this is where the true coelom comes in. But it's also in these other spots as well. So those other rings, they all have a true coelom. So that's why that coelomate differentiation doesn't make any sense, because you can see it scattered throughout these different clades. And so, Lophotrochozoan, Ecdysozoan, give a good shout out and welcome to them. They're the new groups, have been around, a couple decades now. And coelomates aren't really used as file anymore. It's just like, hey, it's your way to describe an organism. Like, hey, what kind of tissues you got? What you got? Which characteristics can you see? It's on those same lines of just being a way to describe them. And this shown here is a lophophore feeding structure and the trochophore larval form. It looks kind of cute, like his little tufts of hair. It doesn't have tufts of hair. Only mammals have hair, but it looks like it. All right. Well, nearing the end here of our first online lecture. So, development. What controls how these things develop? And that's where Hox genes come into play. So, Hox genes help control the development of these different body regions of organisms. Like, what stuff is going to become. So, these genes help to figure out, all right, this is going to be the head end. This is going to be the tail end. This is where all that junk in the middle develops. So, this is where the organs are going to be, the heart, the lungs, all that stuff where it's going to be. This is where we're going to grow some arms, grow some legs, grow some eyes, grow a tail if you got it. And there's evidence that this is shared as in the development of all animals. So for common ancestors of animals, except for sponges, they don't have any. But the other ones have this. And so you can see it's very heavily conserved amongst different types of groups. So you can see the fly, which is Drosophila. You can compare it to mouse and then to humans. And it's pretty similar in a way that you would not expect for things that separated out so long ago. So it's pretty ridiculous. So it's very, very similar. And so they've actually done experiments. They put chicken Hox genes into a fly, and that thing developed perfectly fine. Like, there was no difference. Even though they last shared a common ancestor over 670 million years ago. So that's pretty insane that was able to work, and shows how heavily conserved these things are. So they help just, give them the identity on what these segments will become, just like lay out the blueprint, if you will, for the development, and then other things will go along and build it. All right, that's my dog barking because neighbors are being loud upstairs. But radial symmetry. We can't ignore our radial symmetrical friends. It's not all about bilateral symmetry, right? Radial things matter too. And it's less common, but still cool. It has top and bottom halves as opposed to a front or like a head end or a tail end. So a sea star has a top half and a bottom half, or also called the oral or arboreal end. The body plan is usually more based on like a sphere or cylinder and not like anything else. So it's kind of, that sort of structure that it has to it. And it centers around a central point. So that's where that cylinder comes in. You have that one point and then things branch off from that. And so very much like a sea star, where you have the middle part, the central disc, and then you have the legs that come off of that, or the arms that come off of that. And so these things are usually not super duper active. So they can move, but they're kind of slow, float along the bottom. A sea star has ways to move, but it's not going to win any races or anything. And planktonic means it kind of floats around in the ocean and just drifts. And so that's pretty common with these organisms. And like with jellyfish, that's what they do pretty much. But you also have some that are completely sessile, like corals and anemones. They don't really move a whole lot. And so there's different ways you can breakdown radial symmetry, but excuse me. But this is good enough for us. But note the Hydra, despite having multiple heads on the bottom right, it's not real. But if it was, it would still be bilateral symmetry. Because even though it has all those heads, it still displays more classic bilateral symmetry. But it's fake, so who cares? But it just tempts you. Okay. So radial symmetry, what are some groups you can find this in? Cnidaria and Forest, first one to talk about, a bunch of jellies, and corals, and hydras. So all those things have radial symmetry. And then echinoderms also have it. Now this is weird because echinoderms are right knocking on the door of chordates. So we'll talk about this later on. But they get by this because as an adult, they show radial symmetry. But as a larval form, they show bilateral symmetry. So that means they just adapted and kind of went back, so to speak, to go back to the radial symmetry as an adult. So that's just kind of an interesting change they can have. And that shows that comparison between larval forms and adult forms, talked about earlier, and how that deviates. So like you have a larval form that is very different from the adult form, even in terms of like symmetry, which is pretty cool. All right, so here's radial symmetry. As you can see the difference. A pot is a good example, and also an urchin or anemone, showing. And here's the comparison between the three. Get back here, stop yawning. Oh, my goodness. Okay, asymmetry. I like the picture for asymmetry. It's pretty fantastic. It's just a random blob. That's fantastic. And then like the beetle on the bilateral symmetry, and then the weirdo jellyfish, which is super cool. But that weirdo machine, I can't stop yawning. My goodness. I apologize. Sorry, folks. That asymmetry picture just cracking me up. But you can see it's all based on planes. So where you put the plane down, bilateral symmetries, it's got one spot. Radial, money spots. Asymmetry, you can't put a plane in there, but the thing makes it look like sense. That's the sponges, really. That's their gig. All right, and this leads us to our last real thing, which is tissues. So tissues, there are 4 main types of tissue you can have in animals. Not all found are all in animals, but at max you can have 4 main categories of tissues. I've taken A and P before. This is going to bring back some flashbacks of like, oh boy, microscopes and craziness. But tissues are cool. They give a lot of different properties. So you have cells that come together, form similar functions. You get a bunch of tissues together, you get some pretty cool organs. So let's look at these. As you can see in the top right picture, we have connective tissue, epithelial tissue, muscle tissue, and nervous tissue. And so let's look at some of those. So nervous is made up of, nervous system stuff. So one of the main units of nerves is neurons. So there's many neurons all connected together in long chains that convey electrical signals for movement. And then glial cells are pretty cool. They're accessory cells that aid in the nervous system. So a lot of times they can wrap around neurons. They can serve serve functions like white blood cells to protect neurons. Because inside the central nervous system is kind of weird. A lot of things can cross the blood-brain barrier. So they have to have their own mechanisms to help themselves and deal with problems. So glial cells can play that role. They also play and make sure nerve impulses can transmit faster via these things called myelin sheaths. That's all made possible by glial cells. Muscle, obviously, allows for movement, duh. But you have different types of muscle, right? You have skeletal muscle, which is voluntary, so you can get up, walk out the room, turn off this thing because you're done with this already. Whatever. You also have cardiac muscle, which makes the heart beat over and over again. Otherwise, you die. That's pretty rough. So heart beats by itself. The heart's pretty cool because it has its own electrical signals to generate it. And it's cool. Take A&P, recommended. You learn a lot more about this stuff. And smooth muscle. Smooth muscle also is involuntary. The body kind of just regulates that on its own. You don't have to do it. So this helps move food through your system. So when you eat a meal, food moves through you, right? Smooth muscle makes that possible through a thing called peristalsis, which is rhythmic contracting. Then we have connective tissue, which is a crazy one because you have so many different types of tissues. So cartilage is included in this, which is, it's just like a weaker version of bone, but bone's in this too. And then you have blood, which is a liquid tissue, which is, it's going to be insane. It's really weird. You don't think about blood being a tissue, but it is. And it's the same category with bone, one of the hardest tissues around. Then you also have blood, liquid tissue. What? And then adipose, which is of course fat. And there's many different types like dense, regular, dense, irregular, just various other connective type of tissues that serve a wide variety of functions. And then one of my personal favorites, epithelial, is where it's at. Epithelial is amazing. So this is like your skin and also your internal linings as well. So like in between, like inside your mouth, all the way down into your GI tracts, with your stomach, esophagus, small intestines, large intestines. All that stuff has linings to it, and so that's all epithelial tissue. And so I love epithelial tissue. Skin is amazing, unsung hero, keeps you safe all the time, and at least tries to as best as it can. Things can grow on it, but there's many adaptations on skin to prevent like bacteria and other things from developing on your skin, which is really cool. But yeah, a lot of different types of stuff here in this tissue. So here's some more pictures. Here, so we can see the brain for the nervous system, crazy blood cells, the heart with cardiac muscle, skin on the bottom right, which, I mean, what they're doing isn't a lot of hands touching, which is frowned upon now because of, coronavirus, or COVID-19, or SARS-CoV-2, all kinds of names depending on how fancy you feel like being. Then, of course, we have neurons in the middle picture on the right side, where you have a big blob and the extensions coming off. Oh man, neurons are so cool. And then bones, which are pretty awesome, right? Bones, so skulls. Okay, so that is it for that. I think we've kind of wrapped up tissues and body plans. And so there'll be a little quiz thing I'll put up so you can do a little practice on what is symmetrical, what is not. So check that out, complete that, so you can make sure you get some practice on bilateral versus radial symmetry. But other than that, folks, it was fun, it was real. This is our first adventure of many. So yeah, stay tuned for the next one.